Hello, I'm Professor Sims, and this video is an introduction to writing lab reports for the microbiology lab. So the written assignments for this course, except for bonus assignments, all consist of 4 regular lab reports and 1 semester project report. These assignments are designed not only to help you collect, analyze, and interpret data, but also to strengthen your scientific writing skills. While scientific publications may vary in format depending on the journal or the discipline, several universal elements appear in nearly all reports. The introduction, materials and methods, results, discussion, conclusions, and reference section. The introduction section is where you set up the stage for your report. Here is where you explain why your subject matter is significant and provide important background information to give context. This might include recent findings, examples, or citations from scientific literature. You'll also connect your work to the course or unit learning objectives. These are the goals that you hope to achieve by performing the procedures and analyzing the results. If you have developed a hypothesis, the introduction is also the place to clearly state it, along with how you intended to test or research it. Think of this section as both the roadmap and the justification for the work that you have carried out in lab. When we talk about a hypothesis in science, it's much more than just an educated guess. A hypothesis is an expected result of the experiments that you plan to perform based on the information that you already know. In other words, it's a predicted outcome, or what you think will happen, along with the rationale or reasoning for why you expect that result. A strong hypothesis has 2 key qualities. First, it must be testable. You should be able to design an experiment that could support or reject it. Second, the outcome must be measurable so that your results can provide evidence either for or against your prediction. For example, if you're writing about a demonstration lab or a procedure designed to show you a concept, it would not be appropriate to create a hypothesis. Instead, you would just focus on the learning objectives for those experiments. But if you were running an experiment, say testing bacterial growth before and after hand washing, it would make sense to form a hypothesis. You might predict that the plate inoculated after hand washing will show fewer bacterial species because the detergent in the soap should reduce microbial populations. And that is a testable, evidence-based hypothesis. Finally, remember that a hypothesis is never proven right or wrong. Instead, it is either supported or rejected by the experimental evidence that you gathered. The materials and methods section is where you explain exactly how your experiments were carried out. The goal is to give your reader enough background and detail so that they can understand what you did and, if needed, actually repeat your experiments. In this section, you describe what experimental procedures were performed, how and why those procedures were chosen over other options, how those procedures work, and what kind of information they provide. Many students find it easy to describe what they did in lab, but it can be more challenging to explain why those procedures were appropriate or how they worked to help test your hypothesis or reach your learning goals. That deeper explanation is just as important as the step-by-step description. In fact, I don't want a step-by-step description. What I want is for you to explain what you did in such a way that it makes it clear to the reader why you did it that way and not some other way. And then finally, if you made any modifications, or even if you didn't, you should include in the lab report those changes that were made or clearly state that no changes were necessary. The results section is probably the most straightforward part of the lab reports. This is where you present your observations, data, and analysis of the experiments that you performed. The key here is honesty. Include your true findings, even if they weren't what you expected. Remember, unexpected results can often lead to more learning than results that go exactly according to plan. Your results will often include tables and figures. Every table and figure must be labeled with a number, a title, and a caption. The caption should provide enough detail so that someone, if they were looking only at the figure and its caption, that they would still be able to understand what that figure is showing. Tables are typically used to organize repetitive or numerical data, while figures like graphs or images are better for showing trends or visual outcomes. And no matter which one you use, make sure every table and figure is directly cited in your written reports or in your written results. For example, you might write, as shown in figure 1, to point the reader to the correct data. The discussion section is where you explain the significance of your findings. And it's not just about presenting data, it's about interpreting the data. So here is where you want to make connections between what you observed and what was already known, or what your learning objectives were. So you would start by restating the learning objectives in summary form, and then explain how performing the laboratory procedures and analyzing your results helped you to achieve those objectives. This is also the place to describe any new understanding that you gained about the specimen, the experimental procedures, or even the scientific concepts themselves. This discussion is not about what you did so much as what you learned. Think of this section as the mind of the report, where the intellectual work really happens. And that is in contrast to the conclusion. The conclusion is different from the discussion. While the discussion focuses on what you learned, the conclusion focuses on the procedural and experimental design aspects, almost like the hand of the report. Here is where you're going to clearly state whether your hypothesis was supported or rejected. You also will compare observed results to expected results. Were they similar? Did they differ? It's also important to point out any sources of error that could have influenced your outcomes, and to discuss how significant those errors might be. Finally, if you were to repeat the experiment, what changes would you make to improve the process? You can even suggest possible alternative explanations for your findings when appropriate. So in short, while the discussion interprets your results in terms of learning, the conclusion ties your results back to your hypothesis and the procedures that you used to get there. For some do's and don'ts, do work on the reports incrementally. Work on them as you go, because a lot of what is included in the report can even be completed well before you have done any procedures at all. Do follow the standard format. Templates are provided, and all you need to do is fill in the blanks and format everything correctly. Avoid pronouns, contractions, and slang. Present your organism names correctly using binomial nomenclature rules. And if you don't know, the rules are to always capitalize the genus name, always use lowercase for the species name, and both the genus and species names are to be italicized if typed or underlined if handwritten. Always write in complete sentences and paragraph format. Don't use lists or bullet points. And remember to include enough information in your legends and captions like we discussed before. Don't try to write an entire lab report the day before it's due, because this will mean the difference between getting an A on the report or a C or a D on the report, because it undermines the learning aspect of completing these reports. That is, if you cram it all in one setting, you're not going to get the full benefit intended. And also, there's a lot of time between when you're performing the experiments versus when you get the results, and then the reports cover more than one lab session, so you're bound to forget a lot of the things that we discuss in class if you wait too long to start. Don't include irrelevant or extraneous information. Don't make up results to match what you think should have happened. Don't use vague descriptions. You want to be really specific. And of course, don't plagiarize or use information from a source without properly citing them, giving credit where it's due. Plagiarism is a very big deal. If you're caught plagiarizing, you can be expelled not only from my class, but also the university. It is your responsibility to understand what plagiarism is and how to avoid it. Accidental plagiarism is still plagiarism. I've provided a very nice article in the Moodle site in the lab report module called Avoiding Accidental Plagiarism. Please take the time to read through that before turning in any written work for this course. And then your citations, the full citations go in the reference sections. The reference section. All sources that you use in your lab report must be cited properly in APA format. And this means that each reference needs to include the author's year, title, year of publication, title of the work, and source details, whether it's a textbook, journal, online database, website, video, whatever it is. And then there's two types of citations. You have in-text citations and full citations. The in-text citation goes in line with the text. It should follow the information that is being cited, and it should be in the form of an open parentheses, followed by the first author's last name, a comma, and then the publication year, and close parentheses. A full citation goes in the reference section at the end of the report, and it is going to include all of the authors, the publication year, the title of the work, and the source, whether that be a webpage or book, journal, wherever it came from. In short, the full citation includes the author, year, title, and source, just like the example that is shown here. Authors, year, title, and then source. This is a journal, the volume of that journal, and then these are page numbers. So all references and citation must include an author and a publication year. And this is very important for scientific information as it must come from a reliable source and it must be relatively current. Speaking of reliable sources, reputable sources of information include things like your lab manual, scientific textbooks, peer-reviewed journal articles, my video presentations. Some online sources are also acceptable, but not all. Reliable options include databases like Google Scholar, PubMed, Open University, Medline Plus, and Science.gov. These are just some examples. When you're looking online for sources, a quick tip would be to pay attention to the ending of the website address. .edu and .gov sites are almost always trustworthy. .org can also be good, though sometimes they represent special interest groups. Be cautious with .com and .net sites since they're often commercial or personal websites. And remember to never cite Wikipedia. If you find something useful in Wikipedia, go to the references at the bottom of that page, and then you can find original sources to cite there instead of Wikipedia. The templates from your lab reports can be found in the Moodle module for lab reports. They'll be in a folder called Lab Report Templates. You have a template for all 4 of your lab reports. You would click on those, click on the appropriate one. Lab Report 1 covers lab sessions 1 and 2. Report 2 covers lab sessions 3, 4, and 5. Report 3 includes sessions 6, 7, and 8. And then report 4 covers lab sessions 9 and 10. So that's where you would download the template. And then once you have the template, you see all these gray boxes. That's where you would complete the report. So it's essentially a report that was already written, but there are pieces missing that you need to fill in. So you don't need to rewrite the introduction or anything like that. You're just simply adding to the template. I've also included in the lab reports module a tutorial for importing images to a Word document. If you are struggling with that, you can always let me know. I can demonstrate it in person or over Zoom, but it's pretty straightforward. And that tutorial that I've given you is for multiple versions of Word, and I know that even 2010, that's getting pretty old. So if you're still struggling after reading the tutorial, then just let me know and I'll help you. And then finally, once you are finished with your lab reports, you're going to save it. I recommend putting your name in the file name if possible. It just makes it a whole lot easier for things to not get mixed up or lost. Save it and then you upload it at the assignment link. So the assignment links will look like this. And then you'll see when I grade your lab reports, you'll see the rubric and where you scored for each section. And then I also include feedback for any points that you will miss. So that will help you to not miss the same kind of things in later reports. So you get all your points. Okay, thank you guys for watching, and don't forget to do the reading. Check the description for more videos related to these topics and leave your questions for me in the comments below.